Okay. All right. So last time, on Wednesday, we finished up our discussion of the light-dependent reactions of photosynthesis. Okay? Somebody real quickly summarize the 2 or 3 main events that take place during the light-dependent reactions of photosynthesis. 2 or 3 main events. And the reason why I say 2 or 3, because it really depends on whether you lump some main events together. Yeah?
Water is broken and a photon of energy is used to excite an electron.
Yeah, okay, so you've got some main events. We're splitting water and a photon of light exciting an electron. Absolutely. Those are, I would say that's the same main event, because when the only way to split water is by exciting that electron and stripping it away from there, which makes water then unstable and it falls apart into hydrogen ions and oxygen gas. Yeah. And so that is absolutely one of the main events. We split water by exciting an electron. Allison.
And then the electron bounces between the pigments as well, which is the wavelength.
So the electron, well, the light typically, the electron doesn't bounce around. Light energy will bounce from one pigment molecule to another until it gets to the right wavelength to strip that electron away. Either strip it away from water or take it from a very low energy portion of the photosystem up to a high energy. That's what we see happen in photosystem 1. In photosystem 2, when you hit that right wavelength, it strips an electron away from water. In photosystem 1, when you hit that right wavelength, it excites an electron from a low energy place in photosystem 1 to a high energy place in photosystem 1. So that then it can go from there to ferredoxin and ultimately to the final electron acceptor. Okay? So the really big events that I would say, and again, you can say 2 or 3, I would say the 2 main events are energy production and reduction. Energy production in the form of ATP, which happens during the light-dependent reactions of photosynthesis, and reduction. Ultimately, the reduction of NADP plus into NADPH. Okay? Those are the 2 big events. But again, if you want to separate the reduction into the oxidation of water, right? Because water has to be oxidized in order for ultimately NADP plus to be reduced, then that's fine. Then I think you've got 3 main events. Oxidation of water, energy production in the form of ATP. Well, you can't produce energy, but energy formation, ATP formation, and the reduction of NADP plus. Okay, those are the 3 really big things that happen during the light-dependent reactions. If those don't happen, you cannot go through the light-independent portions of photosynthesis. Okay, and so we talked about on Wednesday the cyclic flow, right? The cyclic flow of electrons rather than the linear flow. Linear flow is when electrons go all the way from water to the final electron acceptor, which is what? NADP plus, it's oxygen in cellular respiration. The final electron acceptor in photosynthesis is NADP plus. Okay? So the linear flow, electrons, again, they go all the way from water to the final electron acceptor, NADP plus. In the cyclic flow, the electrons go from photosystem 1 at that high energy place, the primary acceptor, all the way through something and come back to photosystem 1, now at a really low energy. And then they get excited up to that high energy, the primary electron acceptor, and then they cycle back around, the cyclic flow. In that cyclic flow, we're only accomplishing one of those 2, maybe 3 big events. We're only accomplishing ATP production in the cyclic flow. When the electrons just go from photosystem 1 back to photosystem 1, back to photosystem 1. The only thing we're doing is forming ATP. We're not reducing NADP plus. We're not oxidizing water, which means that we cannot do the rest of photosynthesis. Plants during that time cannot make sugars. What they can do is continue to form ATP. So it's basically a period of just basically forming ATP to fuel cell processes until they can start making sugars again. And a plant can't do that indefinitely because not all portions of a plant are photosynthetic. Not all portions of a plant have photosystems and chloroplasts. Yeah, David.
And one more time, so the plant produces the sugars for itself, right?
Correct.
Okay.
Now we benefit from that.
Okay.
Yes, but the plant produces sugars for itself. Now the beauty of it is the plants always produce more sugars than what they need. And then there's the question of why. And it's like, well, maybe they're adapted for times in which they need stored up sugars. And that's true for some tissues, but not for an adult plant. And so then the question is, well, why? And it's like, well, it's kind of what's necessary in order for an ecosystem to work. And it's like, well, yeah, but the plants can't, natural selection can't select for features in a plant that make the ecosystem function. And so it's like, well, why? Why do plants produce more sugar than they need? It's because that's what's required for life to work, and that's what the designer set up so that the ecosystems work. But anyways, that's another conversation for another class. All right, our last questions, sorry, plural. What are the basic processes of the light-independent reactions? How does the Calvin cycle produce sugars and other molecules? What is the C4 pathway and why is it necessary in some plants? And then where and when does the Calvin cycle happen? Where and when does the Calvin cycle happen? Okay, these are our last 4 questions. Now, the last 2 questions, I will say, are not answered in your textbook. This is material that's been pulled from another textbook. This is the only area of the entire textbook where I feel like it was missing something. So I don't want you to miss it. I don't want you to miss out on it. So I provided it for you. You're welcome. All right, so his first question. What are the basic processes of the light-independent reactions? Okay, so after the light-dependent reactions, we have used water as a product, yes? And we have formed oxygen gas. We've used water as a reactant, not as a product. Wow, that was bad. We've used water as a reactant, and we've formed oxygen gas. So we've accounted for one of our reactants in that overall process of photosynthesis, right? That reaction that never really happens. And we've produced one of our products. But we haven't used carbon dioxide as a reactant, right? And we have not produced sugar. Okay, so those have to happen in our light-independent reactions. So during the light-independent reactions, we take what we made during the light-dependent reactions, that is NADPH, which has some electrons, it's been reduced, and the ATP that we use, and we use these to fix, and following that, to reduce carbon. So to fix carbon means to attach it to something. To fix carbon means to attach it to something. And this may be unfortunate because it's not the way we typically use the word fix. We're like, what's broken in the carbon, right, that needs to be fixed? And you're like, oh, the carbon's so sad because it's not sharing electrons evenly with carbon dioxide. And while that might be true, although the carbon's not sad, it's just would rather have more electrons or spend more time with the electrons. It doesn't really, that's not what we mean by fixing. We're not fixing something that's broken. When we fix carbon, it means to attach it to something. Okay, so first we use the electrons and the energy from ATP to fix carbon and then to reduce it. So the Calvin cycle has 3 phases. The first one is carbon fixation. We take a molecule of carbon dioxide and we attach it to ribulose 1,5-bisphosphate, RUBP. And so a lot of you picked up on that this is a sugar, ribulose is a sugar, and then bisphosphate means 2 phosphates. We've got a phosphate attached to carbon 1, we have a phosphate attached to carbon 5 on this 5-carbon sugar. And so when we attach carbon dioxide to that, we now have a 6-carbon molecule. The enzyme that facilitates that reaction is called rubisco, which stands for RUBP, that should be a capital P. RUBP carboxylase oxygenase, or called rubisco for short. Carboxylase, this is referring to the fact that it can fix carbon dioxide onto RUBP. Oxygenase, an indication that it could also fix something else onto that RUBP, and that's oxygen, and we'll deal with that in a little bit. Okay, so carbon fixation. This part right here is not directly part of reducing carbon. This does not reduce the carbon when you fix this to it. Okay? The reduction comes second. The second part is the reduction of the molecule. Okay, so the second part was once you attach that carbon dioxide to a 5-carbon sugar, you get a 6-carbon molecule that immediately falls apart into 3-PGA, and this gets reduced and further phosphorylated into ultimately G3P, glyceraldehyde 3-phosphate, which is a molecule that we also find during glycolysis. It's a molecule that you get during the first 5 steps of glycolysis. So this phase is the reduction. So you had a quiz question that asked you, how does carbon dioxide get reduced? And some of you talked only about the light-dependent portions, which is okay if you want to talk about the light-dependent portions and how you get oxygen or how you get electrons from the water generating oxygen. But if you don't actually talk about the reduction of carbon in the light-dependent portions, you haven't really addressed that question. Once you get to here, the reduction of carbon is done. Carbon now, once you make 2 molecules of G3P, the reduction of carbon dioxide, it is done. The third step of the Calvin cycle is not to further reduce carbon. The reduction is done at this point. The third step is just to regenerate the RUBP so that we can do it again. Okay? It's similar to the Krebs cycle, how during the Krebs cycle, we fully oxidize the carbon about 2/3 of the way through the cycle. The rest of the cycle is just to restore oxaloacetate so that we can do the cycle again. Okay? So the reduction of carbon is done after this step, once you have 2 molecules of G3P. This step is just, again, it's to restore RUBP so we can do the cycle again. Now, it requires 6 turns of the Calvin cycle to get 1 equivalent of a 6-carbon sugar. It requires us to do the Calvin cycle 6 times. Every time we do the Calvin cycle, we fix 1 molecule of carbon dioxide. How many carbons are in a molecule of carbon dioxide? 1. So it makes sense that we have to do this 6 times to get the equivalent of 1 6-carbon sugar, right? You do this 1 time, you fix 1 carbon. You do it 6 times, you fixed enough carbon to get 1 6-carbon sugar. Maybe glucose, maybe fructose, some other kind of sugar. Some of you on your quiz picked up on that you have to do this multiple times, but some of you wrote you have to do this 3 times to get 1 molecule of, you have to do this 6 times to get 1 molecule of G3P. This is not true. And you don't have to do this 3 times to fix 1 carbon. You do it once to fix 1 carbon. You just have to do it 6 times to fix enough carbon to actually get a sugar, to get the equivalent of a sugar. So if you do this 3 times, fixing 1 carbon each, how many carbons have we fixed if we do it 3 times? 3 carbons, which is 1 molecule of G3P. So if you do it 3 times, you can get 1 molecule of G3P can be used to produce a sugar. Okay? You do this 3 times, you get 1 molecule of G3P that can be used to produce a sugar. The other 5 will be used to restore RUBP. Okay, and I'll show you this in a minute. And so here is the overall reaction of the light-dependent portions of portions of photosynthesis. And this is a reaction that doesn't happen in a single step. This is a reaction that takes place over 2 steps, but ultimately you take 1 molecule of carbon dioxide, 2 molecules of NADPH, which is 4 total electrons, because each one has 2, right? Just like our molecules of NADH in oxidative phosphorylation, or our molecules of FADH2 in oxidative phosphorylation. 3 molecules of ATP, and what we get is 1 molecule of CH2O, basically the equivalent of a 1-carbon sugar. A 1-carbon sugar. But if we do this 3 times, we get a 3-carbon sugar, G3P. Okay, glyceraldehyde 3-phosphate. Okay. That's pretty good. So for every 1 carbon we fix, we need how many ATP? For every 1 carbon we fix and reduce, we need 3 ATP. So if we're going to make a 6-carbon sugar, how many ATP are required? 18, right? 3 for every 1 carbon. If we're going to make a 6-carbon sugar, it requires 18 ATP. How many ATP do you get out of cellular respiration if you're able to do the whole thing? 32, right? You get 2 net out of glycolysis, 2 more out of the Krebs cycle, and then 28 out of oxidative phosphorylation, right? Do you remember that? So for 18 ATP investment, the plant can get 32 ATP out of that sugar molecule. Like, that's so cool. Not only did it get the ATP by harnessing sunlight, so it didn't have to eat something to get that ATP, but it actually gets more ATP out of burning the sugar molecule than it required to make it. And you're like, well, no wonder why they would go through this process of making sugar molecules. Because you're like, why wouldn't they just use the ATP from the light-dependent reactions? And it's because they require less ATP to make the sugar than they actually get out of burning it. You're like, that's so cool. How does that work? And it's just what you can access. Yeah.
So when I was trying to do the question, one thing I was confused on is that I thought G3P had 3 molecule or 3 carbons in it.
It does. Yep. So you can't get a G3P doing the cycle once. If you do the cycle once, you actually generate 2 molecules of G3P. But neither of those can actually be used to make a sugar.
Okay.
You have to do it 3 times. And so if you do it once, you get 2 molecules of G3P. If you do it 3 times, you get 6 molecules of G3P, right? 3 times 2 is 6. And so if you do it 3 times, you get 6 molecules of G3P. One of those can be used to make a 6-carbon sugar. Or one of those can just be left as a 3-carbon molecule of G3P. Yeah.
Okay.
Does that make sense? And then the other 5, so that's 6, one of them can leave. The other 5 go into regenerating RUBP.
Okay.
Yeah. Okay. All right. So here is a visual representation of this. I don't need you to memorize this, although I would say that this diagram here is probably far more helpful for you to be able to understand and describe what's going on during the light dependent or light independent. I keep saying light dependent. The light independent portions, those that don't require light. This diagram, if you could understand this, if you could replicate this, and again, I'm not going to require you to. I'm not going to give you a blank circle and ask you to replicate this on an exam. But if you could do this, this would probably be far more helpful for you than the previous slide. Okay? And so you can see the 3 phases. We're fixing carbon, attaching it onto a 5-carbon sugar to make a 6-carbon molecule that immediately falls apart into 2 3-carbon molecules. Okay? And then that goes through reduction, a 2-step process where we use ATP and we use NADPH to generate 2 molecules of G3P. Okay? And then at this point, we need to regenerate RUBP. We need to regenerate RUBP. Now we have to do this 3 times just to get one of these molecules that can leave. You see how this arrow shows leaving to where we can make some kind of organic molecule like glucose? You have to do it 3 times to get one of these that can leave. And the other 5 will go into regenerating RUBP. If you do it 6 times, you can get 2 molecules of G3P to leave, which is what you need to make glucose. You need 2 molecules of G3P. And that makes sense if you remember, and this is what I try to do so that I remember these numbers, is that it takes 6 molecules of carbon dioxide to make 1 glucose molecule, right? Because in cellular respiration, we take a glucose molecule and we ultimately oxidize it into 6 molecules of carbon dioxide. Yes? So it takes 6 molecules of carbon dioxide to make 1 glucose molecule. Well, if we only fix one molecule of carbon dioxide every one time we do this, how many times do we have to do it? We got to do it 6 times. We got to do it 6 times. Okay? And that's how I remember it. And that's how you should remember it. Because if you try to just remember it otherwise, you're more likely to make a mistake. You may not. You may not, but I think you're more likely to make a mistake. All right? Yeah, Allison.
So the carbon RUBP, did that all originally come from carbon dioxide?
Yeah, it's a kind of a chicken and egg question. I mean, the plants at the time that a plant develops, it will build RUBP. It has enough organic material stored in the seed to build RUBP from before it becomes actively photosynthetic. Because until it becomes photosynthetic, it can't fix carbon dioxide, right? So a plant like a seedling that's still under the soil and hasn't erupted yet, it's obviously not absorbing sunlight, and so it can't go through photosynthesis, and it can't start harvesting carbon dioxide yet. So it'll build it from organic material that it has access to until it can start building it from carbon dioxide. Yeah, that's a good question. But ultimately, I mean, all of the carbon dioxide it's fixing is going into making sugar. And it's rebuilding RUBP from what it already had. Yeah. Okay. Any other questions about this question? Okay. So what I want you to do is this. I want you to work a little bit with those around you. I want you to take the general reaction of photosynthesis. Okay? The general reaction of photosynthesis, which is CO2 plus H2O produces a sugar molecule and oxygen. Okay? I want you to take that general reaction. The first thing I want you to do, working with those around you, I want you to balance it. Okay? Balance that chemical reaction. Then I want you to note which portions happen during the light independent, what happens during the light independent, the light dependent and light independent. And then what I want you to do is I basically, off to the side, I want you to write down what we still need to do following the light dependent portions and the light independent portions. Because still at this point, we don't have glucose yet, right? In the light dependent portions, we generated NADPH and we generated O2 and we generated ATP. Yes? In the light independent portions, we've generated G3P. So we still have some work to do. So again, I want you to take that general formula. I want you to balance it. I want you to note which portions that those half reactions happen in. And then I want you to note what do we still need to do to get glucose. All right?
What's the formula?
The formula? Oh, the equation? Yeah. Oh, yeah, the formula. Somebody left me a box of markers. That's wonderful. CO2 plus H2O produces oh wow. I got scared. C6H12O6 plus O2. So I want you to take that, I want you to balance it. I want you to note, bless you, the half reactions, you're welcome, the half reactions, where they happen, and then I want you to write off to the side, what do we still need to do to make glucose? Because you should not be able to note this being produced in either the light dependent or the light independent. Okay? Take a couple of minutes, work with those around you, and we'll come back together.
The light independent takes the carbon and puts it in the G3P molecule, right? So we need to break that apart and put that carbon.
I got it.
Sounds nice.
6.
Oh, okay. Yeah.
Yeah,
So what's the missing?
that's already 12.
What's the missing?
Alright.
I'm done.
Okay.
So yeah.
Are you sure it isn't, because this is 6 O's?
So that's 12 and 12.
Oh, wait, oops.
I'm done. I'm done with it.
Okay, well now we have to talk about everything else now.
What is it?
Okay, so we talked about how, okay, the light dependent part happens with the water, right, with the
Yeah.
electrons.
The light is independent.
Like, independent. Like, okay.
And then from
My announcement this morning is working. Everybody's getting their genetic stuff in.
And this is also the product
Alrighty.
of the light independent.
Okay, you've had slightly under 2 minutes. Okay, so task number 1 is to balance this. How many molecules of carbon dioxide do we need? 6. How many molecules of water do we need? 6. Well, what did I do here? Did I do this, or did somebody come up and erase it and make this look ugly? Wow, that was terrible. And then this will be required to make 1 molecule of glucose and 6 molecules of oxygen gas. Now at this point, this reaction looks very, very similar to the opposite of the reaction of cellular respiration. And again, neither one of these reactions actually happen. Okay? These are just are summations of processes. Okay? Now this part, this is definitely fixed during the light independent portions. However, we do not make this during the light independent portions. Now this gets converted into this during the light dependent portions. That one we've got figured out. That definitely happens during the light dependent portions, where we take water, we split it, we pull the electrons away from it, and that generates oxygen gas. This, however, happens during the light independent reactions, plus requires additional reactions. And really what it requires is essentially, this is bonus information, but what it requires essentially is to move through reactions 1 through 5 of glycolysis backwards, in reverse. It requires you to move through reactions 1 through 5 in reverse. You're like, wow, it's so weird. Why would they do that? And the answer is because they have to. Because they need something that can be stored as big chains of energy. And G3P isn't going to cut it. You need to get glucose, and then you can string glucose molecules together as a form of energy storage that plants use, which is what? Do you remember this back to our second week? Starch. It actually may have been week 1. Starch. We do the same thing, but we store glucose as glycogen, but you need to get it to glucose. So we also do this, run through reactions 1 through 5 in reverse. It's called gluconeogenesis when we do it. And it's because sometimes you need glucose so that you can string them together to store them as energy. And sometimes you need glucose because that's the only thing certain cells can metabolize. Red blood cells and neurons can only metabolize glucose. They can't metabolize anything else. And if you're not actively eating something that's providing glucose and you've worked through your glycogen stores, which you usually exhaust your glycogen stores in between meals, unless you eat like every 2 hours. Okay? So you're usually out of glycogen in between meals. Going from lunch to dinner is usually enough to get rid of the rest of your glycogen. But you still need glucose. You got a hungry brain and hungry red blood cells. And so you'll do this as well. Run through reactions 1 through 5 in reverse and generate glucose. Plants will do this too so that they could store excess energy and so that they could transfer it around well. All right? Does that make sense? Okay, so this happens during the light-dependent reactions. This to sugar actually requires the light-independent reactions plus some additional reactions. And you don't need to remember what those additional reactions are. You do need to remember that we do not generate glucose in the Calvin cycle. We generate G3P. Okay? We generate G3P in the Calvin cycle. Okay? So if you were asked a question, I don't know, on an exam that may or may not be a week from Friday, to summarize what is made during the light-dependent or the light-independent reactions of photosynthesis, if you were to say glucose, you would lose points because you do not make glucose during the light-independent portions of photosynthesis. You make G3P. Okay? Is that all right? Make sense? You love it? Can you learn to love it? All right. So how does the Calvin cycle produce sugars and other molecules? We've already mentioned a lot of this, so we'll work through this slide relatively quickly. Each turn of the Calvin cycle reduces a single molecule of carbon dioxide. Each turn requires 3 ATP and 2 molecules of NADPH. So therefore, 3 turns require 3 molecules of carbon dioxide, 9 ATP, 6 NADPH, and they generate 6 molecules of G3P every 3 turns. One of those 6 can leave the cycle and be involved in some additional reactions to make glucose or fructose, some other sugar. The other 5 go through a series of reactions requiring 3 additional ATP to regenerate 3 molecules of RUBP. Yeah.
Can the 3GP produced be used directly in the respiration process?
Yes. Yep. Yeah, because we talked about, Davies asked, can the G3P that you produce be used directly in cellular respiration? And the answer is yes, as long as you can get that molecule of G3P into the mitochondrial matrix, where that machinery is. Right now, it's generated in the stroma, in the fluid of the chloroplast. But if you can get it from there into the mitochondrial matrix, yes, absolutely. And it oftentimes will be. So plants can use that G3P to do a number of different things. They can do it to make glucose or fructose or some other sugar. They can use it to make amino acids, to make lipids, to make nucleic acids. So they can use that G3P to make any of our biomolecules. If you remember, when we were talking about cellular respiration, we had a question that said, how do other fuel molecules work in cellular respiration? And we showed, we had this diagram where it's like fats, and you can bring them in here as G3P. Amino acids, you can bring them in here as either the acetyl group in acetyl-CoA, or as G3P. And we didn't do nucleic acids, but they can be brought in there as well. And so you can do the reverse of all of those reactions to make those molecules. See, the beauty of it is we eat organisms to gain access to some amino acids that we can't make ourselves. Okay? So we'll eat plants or animals to access those amino acids. Most plants don't eat other plants and don't eat animals. So they have to make all of their amino acids. And they do so using the product of the Calvin cycle. So it's a bit short-sighted. Well, one, it's just wrong to say glucose is made during the Calvin cycle. But two, it's even a little short-sighted to say that's why G3P is made, is so that you can make glucose. Because that's only one of the things that plants do with G3P. That's only one of the things that plants do with G3P. All right? Do you love it, right? So here's a more detailed diagram. The only reason I show you this is some of you, I know that the way your memory works is that the more details you have, the better you're able to see the big picture. That is not how most people are. So for most of you, I would say, look at this, appreciate you don't need to know it, and just move on. For some of you, look at this, and it's like, okay, knowing this will help me see the big picture. Okay? I get it. You're somebody like my daughter. She can't remember anything unless she knows everything. She literally cannot remember people's names unless she learns their first and last name. You ask her some kids she's played with all school year, and we're going on like 9 months now that they've been in school, and she doesn't know their name unless she knows their full name. It's weird. It's weird. You guys can pray for her. All right. What is the C4 pathway and why is it necessary in some plants? What is the C4 pathway and why is it necessary in some plants? Again, these last two questions, you will not find anything on these in your textbook. You'll not find anything on these questions in your textbook. Yeah, Peyton.
Since they weren't in the textbook, then why were they on the quiz?
Because they're on our slides and you have access to the slides. Yeah, you have access to the slides. Plus, your quizzes, you can look up everything. I think most people on that question used material that was not on the slides either. They looked it up. All right. So Rubisco, what does that enzyme do again? Yeah.
Adds itself to the carbon dioxide.
Yeah, it fixes carbon dioxide onto RUBP. Okay, that's what this enzyme does. This is unique to photosynthetic organisms. Okay, it's unique to photosynthetic organisms. You do not find this enzyme other than like temporarily. If you eat a plant, you're going to find some of this enzyme in your intestines, right? Especially if you eat the green leafy part of a plant. Like, why would I do that? And it's like, because it's a great vehicle for salad dressing, right? And that's really the only reason you eat a salad, right? Because people think it's weird if you dump salad dressing in a bowl and eat it with a spoon. Nobody thinks it's weird if you dump it all over lettuce, right? It's essentially the same thing. I don't know why. Sorry. Actually, I'm not sorry. You're welcome. So, no, man, I'm all lost. Oh, yeah. So you don't find this enzyme. It's not produced by anything other than photosynthetic organisms. And it's activated by NADPH and ATP. So it's an enzyme. Its default state is inactive. It's present, but it's inactivated. It's not fixing anything unless there's an excess of NADPH and an excess of ATP. Okay, and that's a good thing to remember because you'll see what can potentially go wrong. What can potentially go wrong when Rubisco is activated? So Rubisco can fix carbon dioxide, and it does. It does a wonderful job of this, fixing carbon dioxide so that then you can reduce carbon dioxide and ultimately you can generate G3P, which can be used by the plant to do a number of different things. However, it also fixes oxygen gas. And we call this photorespiration. We call this photorespiration when it fixes oxygen gas. Now, in most plants, the amount of carbon dioxide in the tissues far outnumbers the amount of oxygen, and so photorespiration rarely happens. Okay? Rarely happens. And the oxygen concentration in the air is typically pretty low, so any oxygen produced during photosynthesis diffuses out of the plant very quickly, right? It makes sense? The carbon dioxide concentration in the air is typically pretty high, and so as carbon dioxide is being used by the plant tissues, it diffuses into the plant. However, plants that are drought tolerant, they are good at retaining water. When those stomata are open, those openings in the leaves and stems and other places that allow carbon dioxide and oxygen to diffuse, when those are open, water leaves the plant tissues, which is fine as long as you have a lot of water in the soil to replace it. So in really moist areas where the soil is always moist, not a big deal. The plants will leave their stomata open all the time. And sure, water's transpiring out of the plant tissues, but it's just being replaced by water from the soil. But in dry, arid places where there's not a lot of water in the soil, there's no water to replace that water with. And so the plants have to try to retain as much of that water as possible. So they leave those stomata closed during the day when their transpiration rate would be highest. So what this does is it makes it a lot more likely that you're going to get a buildup of oxygen being made during the light-dependent reactions, right? As you're splitting water, and you're going to become very deficient in carbon dioxide. So you have what's called the C4 pathway. This will fix only carbon, and never oxygen. So it doesn't matter if you've got a ton of oxygen present or not, it will only fix carbon. And it fixes carbon much better than even Rubisco can. So it traps all of the carbon dioxide that's present. It does require more energy. And what it does is it attaches carbon dioxide onto oxaloacetate, which is a molecule we saw in the Krebs cycle, where it took the acetyl group. But here it takes carbon dioxide onto oxaloacetate. Then that gets reduced into malate, and that can be stored for a while. Okay, and then when you need carbon dioxide, you can take that malate out and you can oxidize it into pyruvate, which can then be used in the pyruvate oxidation, and you can use that in the Krebs cycle, and also generates carbon dioxide. So it's basically a way of trapping carbon dioxide and storing it up until you can use it later when there's not going to be any oxygen being produced. And when would oxygen not be produced? At night, when there's no sunlight to split water molecules, or in tissues that are too deep for sunlight to penetrate. Okay? That make sense? Okay, so we have some tools that plants can use to avoid fixing oxygen. You may ask the question, why does Rubisco fix oxygen better than it does carbon dioxide? And the answer is we don't know exactly. We don't know exactly. So here's just those reactions. Here's Rubisco when it fixes carbon dioxide. And remember, that 6-carbon molecule it makes immediately breaks apart into 2 molecules of 3-PGA. When that enzyme facilitates fixing oxygen onto RUBP, it makes 1 molecule of 3-PGA and 1 molecule of phosphoglycolate. And so this process, the C4 process, allows plants that keep their stomata closed during the day to still be really efficient at producing sugar and producing anything else that they need to from G3P. So the last question we need to deal with is this one. Where and when does the Calvin cycle happen? Because the Calvin cycle is also known as the 3C cycle, or the C3 cycle happens in all plants, whether they're drought tolerant or not. All plants do the Calvin cycle, or the C3 cycle. But those that have to keep their stomata closed during the day have to also do another cycle. So in most plant species, the C3 and the CAM plants, the light independent and the light dependent, that should say light dependent, I don't know why it says plant dependent. The light independent and light dependent reactions happen in all photosynthetic cells. So in C3 plants and in CAM plants, the C3 cycle happens in all photosynthetic cells. In C4 plants, the Calvin cycle only happens in cells that are deep enough that oxygen is not being produced because light is not penetrating that deep into the tissue. Okay? And light is what splits water molecules and produces oxygen gas, yes? Yes. So in C4 plants, what they've done is they only do the Calvin cycle where Rubisco is. They only produce Rubisco in cells that are too deep for sunlight to penetrate. So they're not generating any oxygen gas. Therefore, you're getting very little oxygen gas present. And so Rubisco is not going to fix oxygen gas rather than carbon dioxide. Now in C3 and C4 plants, carbon fixation and the Calvin cycle happen during the day. Okay, so in C3 plants and in C4 plants, carbon fixation and the Calvin cycle happen during the day. Even when the stomata are closed during the day, in C4 plants, they're still doing the Calvin cycle during the day, and fixing carbon just in a tissue deep enough that sunlight can't penetrate. So those tissues, it's equivalent to being at night for them, because there's no sunlight penetrating. In CAM plants, which CAM stands for crassulacean acid metabolism, and if you never remember that phrase, I don't care. I do not care whether you can reproduce that. I just wanted to give it to you. So in CAM plants, they still do the C4 pathway where they fix carbon onto oxaloacetate and then reduce that into malate, but that process only happens at night. At night, when the stomata are open, carbon dioxide is fixed onto oxaloacetate, which then gets reduced into malate, and all of that malate is stored up in the central vacuole. Because CAM plants don't do this C3 pathway at night. They fix carbon at night, but they don't do the C3 pathway at night, only during the day. So during the day, the malate will move into the chloroplasts, get reduced into pyruvate and carbon dioxide, and increase the amount of carbon dioxide present in the cell, making it very unlikely that the rubisco will fix oxygen rather than carbon dioxide, because there's plenty of carbon dioxide. So all of these strategies, and this right here, this is a really helpful slide. It's just a really helpful slide. And the reason why it's helpful is because it shows you the various strategies that plants employ to ensure that their photosynthetic processes are as efficient as possible. That their photosynthetic processes are as efficient as possible. And so maximum efficiency is leave the stomata open all the time, right? As long as you can physically accomplish that. If you're going to lose so much water and you can't replenish it, then that just doesn't work for some plants, right? And so some plants, that's not an option. And so that's why we say as efficient as possible. So C4 plants and CAM plants are not as efficient. So if you take a C4 plant, a drought-tolerant plant, I don't even know if this for sure is a C4 plant, but I'm going to say something like a desert tree, and you plant that in a really moist, arid place, it's going to be completely outcompeted by a C3 plant because it's not as efficient. It's not as efficient. But a C3 plant oftentimes is not going to survive in a dry, arid place unless you water it a ton. And you all, those of you that grew up in the desert, you know what it takes to keep plants that are adapted for moist environments alive because they look good. It takes a lot of water. It takes a lot of water. Sometimes it's worth it because the plants look good. So here's a C3 leaf. Notice we have some bundle sheath cells, but they don't have many chloroplasts. That's because very little light penetrates down to that point. But now we look at a C4 plant, and they only do the Calvin cycle in bundle sheath plants. And so they need a ton of chloroplasts to make that happen, even though there's very little light penetrating here. So there's very little of the light-dependent reactions happening, but that's where all the light-independent reactions are happening, in the stroma of these chloroplasts. Okay? So they have a lot more chloroplasts there. A C3 plant would look just like a CAM plant would look just like a C3 plant. And then you can see these C4 pathways and the CAM pathways, they're virtually identical, except for whether they happen in different cells or they happen at different periods of the day. So CAM plant, their strategy is don't separate the C3 cycle and carbon fixation, but separate them based on time and not on location. C4 plant is separate the location, but not the time. All right? We're out of time. Enjoy the rest of your Monday. Those of you that have lab at 1:55, I'll see you in a little bit over an hour. And I think you'll like lab today. I think you'll like it. I'll like it.